Hello, my name is Banele Methula. I am an ambassador at Nabio Consulting. My talk for the day is titled Theranostic Superparamagnetic Iron Oxide Nanoparticles, Transmembrane Transportation as Drug Delivery System. The development of biology and medical science through advances in material science as well as in nanotechnology has introduced the use of nanoparticles as drug carriers in cancer diagnostics as well as therapeutic strategies. One of the most fundamental aspects that you need to look into when designing a drug carrier is the process that warrants internalization of molecules or solids. This process is called endocytosis. Pictured here are different kinds of nanoparticles that are used as drug carriers as well as their endocytosis pathways. We have 4 properties that help determine the pathway. That is the size of the nanoparticle, the surface area, the type of cells that are being targeted, for example, macrophage cells, as well as the activation status of the cell. For example, we have dendrimers that use the caveolin-mediated endocytosis pathway, as well as MSNs that use the clathrin-mediated endocytosis pathway. But our focus for the day is on SPIONs, superparamagnetic iron oxide nanoparticles. They use the scavenger-mediated phagocytosis pathway, as well as the pinocytosis pathway. This is a typical structure of a superparamagnetic iron oxide nanoparticle. It is coated with a hydrophilic shell that could be from a polyethylene glycol polymer or a polysaccharide. The hydrophilic shell prevents the nanoparticle from aggregation. It facilitates controlled drug release. It extends the half-life of our nanoparticle, as well as it enables easy binding of functional groups. And then located on the surface of, and located on our surface coating are targeting ligands. It could be an aptamer, an antibody, a peptide, or a small molecule. What targeting ligands do is they enable for receptor-based active targeting delivery strategy, tumor cell honing, nuclear localization, intercellular organelle localization. The ionic core enables for MRI contrast, hyperthermia therapy, as well as controlled drug release. Superparamagnetic iron oxide nanoparticle incorporates genetic material, imaging agents, as well as drugs that could be actively or passively targeting to specific locations within the body. Before a successful engulfing of an active delivery system into a cell, it must first bind with specific receptors that are located on the plasma membrane. I will demonstrate this using the lock and key model. First, we have figure X, which is our targeting ligand, going in to bind with figure Y, which is the receptor that is located on the plasma membrane. And then we have a successful XY figure, which is the successful binding of our targeting ligand as well as the receptor. Superparamagnetic iron oxide use 2 types of endocytosis pathways, that is phagocytosis. Phagocytosis is a process where receptors that are in the cell membrane bind with ligand molecules that are located on our SPIONs, then the cell membrane progressively engulfs the target. Phagocytosis has different kinds of receptors. We have, for example, there are mannose receptors as well as macrophage interins. In the case of superparamagnetic iron oxide nanoparticles, there are scavenger receptors. They bind a variety of microbes in addition to modified low-density lipoprotein particles. The second endocytosis pathway is pinocytosis, which is a process of ingestion of liquid droplets. The steps involved in pinocytosis are, we first have a molecule in the extracellular fluid binding to the cell membrane, and then there is a trigger on the cell membrane to create a fold around the fluid containing the molecules that are to be ingested. And then we have the cell membrane invaginate, that is, it folds back on itself to create a pouch. And then this pouch is then pinched off at the cell membrane and then migrates into the cytosol of the cell. We have other different endocytosis pathways. For interest's sake, I am going to tell you about them. It's the first is micropinocytosis, which is the ingestion of larger molecules, followed by a clefrin-mediated endocytosis pathway. That is, we have clefrin proteins that bond and form cages around the molecules that are to be ingested. And then we have a caveolin-mediated endocytosis pathway. Here we have a caveolin integral membrane protein that serves as a collection piece to gather specific molecules. And last but not least, we have a clefrin and caveolin independent endocytosis pathway. An example of the medical application of SPIONs is in cancer therapy, where they are first used as MRI detection agents in diagnostics, and then they're also used for hypothermia therapy, where the spines oscillate and then they transform electromagnetic energy into heat. And then there's an increase in the temperature at the cancer tissue, which induces the death of cancer cells. Superparamagnetic iron oxide nanoparticles as drug carriers are substrates that are used in the process of drug delivery that enable for controlled release, which provides prolonged delivery of drugs while maintaining its blood concentration within therapeutic limits. SPIONs, or supermagnetic iron oxide nanoparticles, enable for a receptor-based active targeting delivery system. The advantages of this system are they increase patients' compliance, they enable for non-invasive routes of administration, they minimize local and systematic side effects, they reduce toxicity profile, and last but not least, they enhance the bioavailability of drugs. I thank you for your time.